Hi everyone, welcome to my channel Pure Biology, and today I come with another video related to immunology. And before going to this topic, I want to say that I have already uploaded different topics related to immunology in my channel. So please you check out all those videos, and obviously this one, and obviously you connect with my channel for the growth of this channel. And I say thank you to all of you who have already subscribed my channel. Okay, so today's topic is antigen. Okay, so what is antigen? Antigen is actually adaptive immune responses arise as a result of exposure to foreign compounds. What is called antigen? Adaptive immune responses. Okay, two kinds of immune responses are there. So what is adaptive immune response? I will explain in another video. Okay, so today is that this adaptive immune response that arises as a result of exposure to foreign compound. That means when any foreign compound that enters in our body, in that situation immune response, adaptive immune response that arises. Okay, and the compound, the foreign compound that is responsible for the inducing or induction of the immune response, this is called the antigen. Okay, this compound is called the antigen. So this compound that evokes the response or induce the response is referred to as antigen. Okay, and a term initially coined due to the ability of these compounds to cause antibody responses to be generated. Same thing, the compound that arise that induce the antibody responses, that is also you call the antigen. Okay, and generally now it is defined as adaptive immune response that arise due to some foreign compounds. These are called the antigens. Okay, both are same. It is specifically said that antibody responses that arise, and here this is stated that the immune responses arise. Clear? Next, what is the antigenicity? The antigenicity is that the ability of a compound to bind with specifically an antibody or a TCR. TCR means T cell receptor. This is known as the antigenicity. That means antigenicity is the capability, you can say, or the ability you can say, that is the actual ability of the antigen. Here it is compound, say that, or the foreign compound. But the ability of the antigen, what is the ability? To bind with specific antibody. Okay, the ability of antigen to bind with specific antibody or TCR, this is called the antigenicity. Clear? Next one is the immunogenicity. What is called the immunogenicity? When the immunogen, any agent that is what is called first of all the immunogen, it is an agent that is capable of inducing the immune response. Okay, this is known as the immunogen. Clear? What is called the immunogen? That is the agent that induce the immune response. This is called the immunogen. And what is the immunogenicity? Its capability or its ability to induce the immune response is called the immunogenicity. Clear? Next, the allergenicity. What is the allergenicity? It is the ability of an antigen to induce various types of humoral and cell mediated immune responses and that have allergic manifestation. That means here allergenicity means the ability of an antigen that induce humoral and cell mediated immune responses. Okay, and I will explain humoral and cell-mediated in another video. Okay. So, two types of immune responses arise due to the activity of an antigen is called the allergenicity. When allergic manifestation is occurring due to the arising of immune responses. Clear? Then it is called the allergenicity. Okay. The last one is the tolerogenicity. It is here the ability of the antigen to induce either humoral or cell-mediated. Okay, either humoral or cell-mediated. So here it is stated that to induce the specific immunologic non-responsiveness either in humoral and the cell-mediated branch. So what is this? The ability of an antigen to induce specific immunologic non-responsiveness. Specific immunologic non-responsiveness. Either it is maybe humoral or maybe cell-mediated. Clear? So these are the terms related to antigen. Okay, so we actually have to learn antigen because it is what kind of foreign particle that induce our immune system or specific antibody bind with this type of antigen. What is this? So that's why we have to learn the antigen. Now we will learn that the requirement of the immunogenicity. That means when this immunogenicity or our immune system become active. Okay, so there are some reasons or you can say some features of antigens are there that induce our immune system. Okay, just foreign particle attack in our body and immune system arise, no, that is not possible. There are some requirements that is very important. We have to know that when our immune system becomes arise, okay, due to the attack of antigen. Okay, now we will learn that thing. Okay, these are the points, requirements of the immunogenicity. First one, the foreignness. Antigen must be foreign particles. Okay, antigen must be foreign particles because if antigen is not a foreign particles, then it is not eliminated by the adaptive immune system. Okay, so it must be eliminated in the to the host with respect to the host. Okay, with respect to the host. Second one is the size. If the size is very small, that means the molecular weight is lesser than 1000 Dalton, then it is not immunogenic. Okay, not immunogenic. Those in some cases when the molecular weight remain between the 1000, sorry for the disturbance. Okay, so if the molecular weight that remains between 1000 and 6000 Dalton, then this may or may not be immunogenic. May be immunogenic or may not be immunogenic. Naturally, immunogenic is not possible because here the molecular weight that remains between 1000 and 6000 Dalton. But sometimes it may be immunogenic or it stimulate or induce the immune system. And the last one, when the molecular weight that is greater than 6000 Dalton, then obviously these are immunogenic. Okay, these are immunogenic. So, example is albumin when the molecular weight is greater than 6000. Aspirin, whose molecular weight example is aspirin when the molecular weight is lesser than 1000, and insulin when the molecular weight remains between 1000 to 6000. So what is very important, the antigen that induce the immune system that have must molecular weight that is greater than 6000 Dalton. Okay, greater than 6000 Dalton. Third one is the chemical complexity. Very important, that is the chemical complexity. Greater the degree of complexity of the protein, the more vigorous will be the immune response. Okay, because protein particles, they have more chemical complexity. That's why they have the ability to induce the immune system. Clear? And in case of carbohydrates, these are also immunogenic when they have the complex polysaccharide structure. Okay, they have complex polysaccharide structure or a part of complex molecules. Example is that glycoprotein. And the nucleic acid and lipids, they are also immunogenic when they are conjugated to the protein carriers. So what is seen here? That is, the protein is very important. Okay, because protein that has more chemical complexity. Okay, that's why when protein particle is present, if this is singly or conjugated with the carbohydrates or lipids or nucleic acids, so in that situation these particles are generally immunogenic. Okay, these are immunogenic and obviously induce the immune system. Clear? The last one, not last one, this is the fourth point, the dosage. Okay, and here the space is very short, so in this point I have written a very small part. Remaining part I just say you in the next part. Okay, or next phase. So dosage and the route of administration. Dosage and the route of administration. What is the dosage? That means the insufficient dose, okay, of immunogen may not stimulate the immune response. Okay, insufficient dose, that means not the sufficient, not greater, not lesser. Sufficient dose is very important. If insufficient dose of antigen is injected or is entered in our body or host body, in that situation they haven't capability to stimulate the immune response. Okay, why? Because they cannot produce more lymphocytes or enough lymphocytes. Clear? And or such dose generally renders the responding cells unresponsiveness or creates the cells unresponsiveness. Clear? So what is that? That is, insufficient dose of immunogen is not important for stimulating the immune response. Why? Either these are not enough to produce enough lymphocytes, or generally responding cells, these are become unresponsiveness. Okay, due to the or this shows unresponsiveness. Okay, the responding cells, they show the unresponsiveness due to the entering of insufficient dose of antigen. Clear? So let's continue. Its part is obviously there. So continue. So fourth point is dosage and the route of administration. So this is continuing from here and it is ended here. Okay. So here the first, here we what learned here, that the amount is very important. So not only amount, the number of doses also important. Okay, number of doses also important. And the second one is the route of administration. Route of administration also affects, because this determines that which organs and cell population will be involved. Okay, so that's why the route of administration is also very important. So there are common routes are, one thing is intravenous, that means the entry of antigen is occurring through the vein. Second one is intradermal, that means through skin. Subcutaneous, that means through beneath the skin. Okay, and intramuscular, okay, that is through muscle. Clear? So what are the common routes? Intravenous, intradermal, subcutaneous and intramuscular. From among these routes, which is very important route that stimulates easily the immune system, that is the subcutaneous route. Okay, subcutaneous route or beneath the skin, that is. This is very important route that stimulates strongestly the immune system or immune response. Clear? The fifth and last point is the degradability. Degradability means the degradation of the antigen. So generally, humoral and cell mediated both require, these are two immune system or the type of immune system that require the interaction of Th cells, okay, with the antigen. In both situations, the Th cells very important, which is added with the antigen, okay, and that has been phagocytosed. So first recognized, then binding with antigen, and last one is the phagocytosis process, okay. And then this is possible where on the surface of the macrophage or antigen presenting cells, that is APC, antigen presenting cells, okay. So this phagocytosis process is occurring where the antigen presenting cells or the cell surface of the macrophage. And here one thing is very important, that hydrolytic the enzymes of the macrophage generally degrade the polymers of the L-amino acid, but these haven't any capability to degrade the D-amino acid. Okay, it can only degrade the L-amino acid but not D-amino acid. And obviously APC has no capability for degradation in case of the immunogens are very poor or weak. Okay, so these are few important points that are required for the immunogenicity. Okay, so here we have learned that about antigen and what features must be there in the antigen which induce the immune system of or stimulates the immune systems of our body. So this is the topic and of antigen, and I think you all understood what is antigen and some terms related to antigen and obviously the features of antigens. Clear? So in the next video I will describe another topic related to immunology. So continue with me.